be incorporated into preclinical screening, and actually for many biotech and pharmaceutical companies, it might actually preclude compounds that at the moment with the assays that they're using are proceeding forward?
Yes, I think it really has the potential, right? So you can see here that our technology really can do real-time detection, generation of those biomarkers related to drug toxicity, right? So we can do real-time so that we can save the mice used for drug toxicity studies, right? So normally in pharmaceutical companies, what they do is they use in vitro assays. Okay, they use in vitro assays to do the screening, but they do it in culture dish. There's many facts injected, right? So if you inject into mice, there are many other factors you need to consider, right? So for our imaging technology, we can really test the toxicology in animal and read out the toxic potential in real time. So I think it's a more precise technology to evaluate the toxic potential for any leading compounds before they enter into clinical trials. Yeah.
Great. Perfect. Okay. Thank you very much, Kenny.
Okay. Thank you.
Okay, we're going to move over to our last speaker in this session, who's going to be presenting their research. So last, but certainly by no means least, we have a talk that is going to provide an intriguing new indication for a drug target class that until now has been associated with cancer. So our speaker is Nam-Hoon Kwon, who is a team leader and senior researcher at Biocon at SNU in Seoul. And Nam-Hoon will be talking us today about the role of a lysyl tRNA synthetase in pulmonary arterial hypertension. Over to you, Nam-Hoon.
Thank you for the introduction. It's my pleasure to have an opportunity to present my study here. My topic is a novel approach for pulmonary arterial hypertension. I majored in life science in Seoul National University. After graduation, I wanted to become a veterinarian, so I was transferred to veterinary medical school. When I was a PhD student, my professor sent me to University of Idaho. It was a turning point in my career. At that time, I screened a bunch of proteins, and I suddenly encountered the tRNA synthetase and got interested in these proteins. tRNA synthetase are known as housekeeping enzymes, but I observed that they play important roles in response to environmental stimuli. So after coming back to Korea and getting my PhD, I joined Professor Sung-Hoon Kim's lab because he is a world-renowned researcher of tRNA synthetase. Since then, I've been working with Professor Sung-Hoon Kim, and now I'm in charge of practical development project in Medicinal Bioconvergence Research Center. We call it Biocon. And I'm also working as a research professor at Seoul National University. My interest is the study of MOA of novel targets in disease, especially tRNA synthetase. And also I'm interested in the early drug development, such as assay and screening development. Recently, I'm working for developing diagnostic tools and therapeutic agents targeting cancer and rare disease. As such, our Institute Biocon is focuses on unique target protein, tRNA synthetase. There are 20 tRNA synthetase, ARS, and 3 cofactors. They are all essential for translation and cell viability. And they are also rapid sensors and responders, and each of the ARS has unique role in different situations. So we have many first-in-class pipelines for cancer, epilepsy, and diagnostic markers and angiogenesis, and sepsis and infections. Today, I'd like to talk about the pulmonary arterial hypertension and KRS today. pulmonary arterial hypertension is a WHO disease classification group 1 disease, and there are several subtypes, kind of idiopathic, heritable, and associated PAH. In the PAH, blood vessels get injured by several factors, and then the vascular deformation proceeds in the following order, such as vasoconstriction, inflammation and remodeling, and plexiform and thrombolysis. These vascular dysfunction lead to blood pressure increase and finally induce right ventricular dysfunction. So PAH is a rare disease with a prevalence with 15 to 50 cases out of 1 million. But the 3-year survival after diagnosis is less than 50%. The current problem in the treatment is that there is no drugs to eliminate the cause of PAH. Current vasodilation and blood pressure lowering drugs are palliative in nature. Only the prostacyclin can increase the survival rates of patients, but it is considered as the final stage of treatment due to the side effects that can lead to death. There is also a limit in improving the quality of life because many PH drugs require catheter insertion, inhalation device, or frequent administration. Also, there is a restriction for the concomitant administration because of the drug interaction and CYP inhibition. In conclusion, there is no drugs. We can eliminate the underlying cause of PAH without side effects while increasing the survival rate of patients. The market of PAH can be divided into 4 major markets: North America, Europe, Asia Pacific, and other areas. Among them, North America is the largest market, accounting for 45%. The PH market is expected to reach $4.75 billion in 2024 from the $3.45 billion in 2014. As shown in this graph, there are 3 major drug categories based on the working mechanism. And among them, the ERA is expected to lead the market in next few years. Interestingly, the PAH is a rare disease, but relatively many new drugs are being developed, showing that 71% of global R&D pipelines is first in class. It is an example to show how high demand for new drugs is in PAH. Many various pre-existing factors are involved in the PAH, and such as genetic factor and infection and related disease. And also immune response around the endothelial cell damage is another important factor for the PAH. Chronic inflammation activates fibroblasts and myofibroblasts, and it induces the fibrosis in the damaged area. Among the immune cells, monocyte and macrophage are preferentially observed around the blood vessel, and after that, smooth muscle hypertrophy, blood pressure increase, and vascular remodeling are observed. So the infiltration of monocyte and macrophage are considered as the most important factors in PAH. Interestingly, the extravasation of monocyte and macrophage requires laminin alpha-4. Our team also investigated several laminin receptors and found that the migration of monocyte and macrophage requires and very specific to laminin alpha-4, especially laminin 421 type. At this point, I'd like to introduce lysyl tRNA synthetase, KRS. As I mentioned before, our institute have investigated the role of tRNA synthetase in disease. Among them, KRS is phosphorylated under the laminin signal, and it exits from the multi-tRNA synthetase complex in the cytosol, and it goes to the cell membrane and stabilizing the laminin receptor and induce cell migration. We have two approaches, compound and an antibody, to inhibit the KRAS-dependent cell migration. Our compound inhibits KRAS in two ways. First, it induces a conformational change of the KRAS, moving from cytosol to the membrane to block the membrane translocation. Second, our compound inhibits the direct interaction between KRAS and laminin receptor. When KRAS is localized to the membrane, the N-terminal domain of KRAS is exposed outside of the cell, and our antibody recognizes the N-term domain of KRAS and inhibit the KRAS-dependent cell migration. As you can see here, our compound and our antibody significantly inhibited the monocyte macrophage migration, which is dependent to laminin 421 type. So we applied our agent for PAH. We administered our compound to the rat, which suffers from the PAH induced by monocrotaline. As you can see here, our compound reduced the right ventricular and the systolic pressure, and the effect is almost the same with the sildenafil, the positive control. We also confirmed that our compound blocked the monocyte macrophage infiltration to the lungs. We also injected our antibody to the PAH animal model. As you can see here, our antibody significantly relieved the symptom of PAH, and our antibody also blocked the migration of monocyte and macrophage to the lungs. All these data shows that our KRAS inhibitor in the form of compound and antibody can be a new therapeutic agent for PAH. We believe that our KRAS inhibitor have the potential as a new PAH therapeutics. Our KRAS inhibitors can inhibit the monocyte and macrophage infiltration to the lung, and it is the main factor of the PAH pathology. And our agents are clearly distinguished from the current palliative drugs in action mechanism, so we think our agent can be co-administered together with the current drug, generating a synergy effect. Another important merit of our antibody is that there was no abnormal signs observed in the single-dose tox test, where even when treated with at the concentration 100 times higher than the effective concentration. So we have registered and filed a patent for the KRAS compound and antibody, so there are no patent issue in the developmental stage. And we are further optimizing our compound, and we are preparing preclinical trial for IND filing with our antibody. So our plan is to analyze the physicochemical characteristics of our antibody and to develop a CMC process to produce toxicology-grade drugs for preclinical trial. At the same time, we will confirm our antibody efficacy in another PAH model, and we will do the non-GLP tox study. Now we are applying for the Korean Drug Development Fund project, and we will initiate the research by spinning off a company which carry out the preclinical and clinical trial. And now we are looking for a partner to cooperate with us. In the PAH pipeline I have introduced, there are another many first-in-class pipelines in Biocon. If you are interested in the PAH project and other first-in-class targets in Biocon, please contact me. Thank you very much.
Great, thank you very much, Nam-Hoon. So I guess, you know, as a new target class, the question will always come up. You know, given that we've been thinking about these again as oncology agents. How concerned are you about toxicities and using these types of therapies as a kind of chronic treatment for PAH?
Yeah, that's a good question. Actually, if we directly inhibit the laminin receptor, that it is very toxic because laminin signal is very important for the normal cell growth as well as normal cell migration. But KRAS-mediated migration is very specific to laminin 421 type, and our antibody has 100 times safer when treated in the effective dose. So we are trying the toxicology study more, but I believe that our antibody is good for the clinical treatment, for the chronic treatment, I think, yeah.
A work in progress. Thank you very much. So if we could just acknowledge all of the faculty that we've had. What we wanted to do with this last talk, we heard at the beginning of the session, Salvo gave us his view of the myriad challenges that we all face. And you've just heard like 6 projects that are all in very different areas. They all pose us, very different challenges, and they will need kind of different solutions in each case. And what we wanted to do before we wrap things up is hear from another really unique view, somebody who has a kind of bird's eye perspective of all of these challenges that we face. He has expertise working in both academic research and working in both industry. He's served on boards of biotech companies. He's worked as head of academic relations at Roche. And now he has his own company, Haystack Sciences. So I'd like to invite Juan Carlos Lopez up here to present his thoughts.
Thanks, Andy. Fingers crossed. Thanks very much, Andy, for the introduction. Thanks very much, Salvo, for the invitation. And I want to start by thanking all the speakers for joining us today. The conditions were somewhat adverse, and I want to severely thank you for that. I think it's been challenging, but I've been very impressed by the science that you presented. So thank you very much for that. I'm not going to talk for an hour, I don't think. I don't think anybody's in a position to put up with an hour of me after such a long day. So I'm going to try to be much faster in what I have to say, which is informed by my time at Nature, where I met Andy and where we started working on the Science Cafe, and our interest in what is it that it takes for a discovery to become a company or a viable commercial proposition. Andy and I have been thinking about this for a long time. Then I moved to Roche, where I thought about it from a different perspective. And after I left Roche, I've kept on thinking about it. And I know that I know finally what is it that it takes, but I've been learning some things along the way, which I hope to share with you during the next few minutes. So let me start by saying something that will be obvious to most of you, and it's the fact that academia and industry must work together. There's no other way around it. And it's been already several years since the pharmaceutical industry declared its unending love for academics. The pharmaceutical industry is very interested in what's happening in academic centers, and there are different reasons for that. On the one hand, they want access to new biology, because there's a lot of biology out there, and the amount of research and all the pharmas combined is very small compared to all the biology that people discover every week. They also want access to new technologies, new ways of doing things that they didn't know before. Today we heard about several of those technologies, in fact. So The pharma industry is very interested in that. They also are interested in having access to expertise. Yeah, they may not want your new target, but if you're the world leader in pulmonary arterial hypertension or in Alzheimer's disease, they want to have access to you as an advisor or as a consultant. And they also save money by working with academics, right? It's always easier and cheaper to have a partnership with an academic center than to actually hire scientists to come and bring that expertise in-house. That's not, financially, that doesn't make a lot of sense. So there are many reasons why academia and industry are destined to work together. Now, so what exactly is it that that industry wants from academics, right? So I already explained some of the reasons, but when you ask a pharma company why, what do you want from an academic group? What is it that they really want, right? And the first thing that you will hear is the following. Oh, we want quality work, or we want novel science. Yeah, so that's what any pharma executive would tell you. We want quality and novelty. What exactly that means is not exactly clear, at least to me anyway, right? What exactly is quality and what exactly is novelty? I'm very critical of these two terms, as you will see in a minute, because if you think about it, and just look routinely at a bunch of scientific papers, these two words, critical and novel, are the most abused words in the scientific literature. Everything is critical. Everything is critical. Every molecule is critical to every disease you can think of. And all of that is always novel, right? In my opinion, if everything were critical, we would have cured every disease already, right? But we haven't. So what exactly does that really mean? So another problem that arises with this is the question of reproducibility, right? So everything is critical, but when you try to reproduce it in an industry lab, the numbers that you hear is that 10% of things reproduce, 20% of things. So it's not really great how easily it is to reproduce something that led to a Nature paper when you want to really translate it into an industry lab. And this doesn't mean that there's fraud or that there's sloppiness. It just means that biology is quite complex and that what works in a certain system may not work in a slightly different system. So that comes with the territory. The other problem is a problem of prioritization, right? So if everything is critical for every disease, how do we choose priorities, right? So we have 5 new targets to treat asthma. How do we go about choosing the one target that we need to really follow up on? I'm in a pharma company and I want to launch a discovery program on the best target. How do I go about choosing the best target out of these 5? The answer to that is that it's quite often, it depends who shouts the loudest, right? Who is the academic that has the most clout, right? And that I'm happy to believe in, yeah, which is a pretty poor way of choosing if you think about it. But that's just the fact. I trust this key opinion leader, I'm gonna go ahead with him. So the process, as you can see, is very inefficient. The process is very inefficient. And part of this inefficiency is because the incentives from each of the different stakeholders are misaligned. In other words, what is important for an academic is not necessarily what is important for a pharma company or for an investor. And here I list very briefly the different incentives so that you see that everybody is pulling in a different direction. So for academics, quite often what they care the most about is advancing in their career. If I'm a postdoc, I want to be an assistant prof. If I'm an assistant prof, I want to become an associate professor. And if I'm an associate professor, I want to become full professor. Right? That's my main goal. I want funding, I want recognition, I want awards. And there was this article published several years ago in the New York Times that referred to scientists as small business owners. Right? You own a small business where what you produce is research. You produce results that you sell to another group of scientists, and they buy it, so they give you more grants. so that you can produce more data that you go back and sell back to the same people. So it's very unfortunate, but that's the reality of academia for a lot of people. For foundations, increasingly the concern is moving the needle in terms of developing cures, as opposed to, here's some money, go and publish some papers. There are fewer foundations that are happy just to give you money. They want to say, so how are you helping the patients with this money I'm giving you? So it's a different priority. For governments, yeah, they want their population to be healthier, but they also want economic growth. They want to create jobs, right? And that's important, but it's a different incentive. For investors and venture capitalists, normally it's a financial question, right? What is my return on investment? What are my multiples? Is this a 5x or a 10x? And what is my exit? Who's buying this when we're done with this investment? So here the incentive is really financial. For people in pharma, quite often is how many deals are we going to do this year? What is my corporate target, right? Because if my target is 3 deals per year and I haven't got a single deal, I need to do deals. So it's a different incentive altogether. And for the pharma industry at large, there's this dilemma of reaching a certain milestone versus is this really the truth that we say that we're interested in. In other words, let's imagine that the target for a pharma company is to advance 5 molecules this year from preclinical development into a phase 1 trial. So whoever has this corporate goal is going to have to meet this goal. And they may not be the